move on now to look at the types of adrenergic receptors. And just to remind ourselves here, the adrenergic receptors are those that are only associated with the sympathetic system. And these receptors only bind norepinephrine, epinephrine, or the hormone version of norepinephrine, okay? So there are two main classes of adrenergic receptors. There are alpha receptors and beta receptors. Each of these two general types have further subdivisions. So there are subclasses associated with alpha and beta receptors, and they are all coupled to G proteins, meaning that they are all metabotropic or all slow receptors, okay? So they're gonna have some type of second messenger involved to bring about their cellular response. They're also going to have a different type of response depending on where that tissue is, because these receptors, as I mentioned before, are customizable. So exactly where the receptor is going to determine rather what response is brought about. So under our alpha receptors, we've got alpha 1 and alpha 2. And then under our beta receptors, we've got beta 1, beta 2, beta 3. And I'll talk about the differences. We'll go through the differences of each of those. So unique properties to alpha adrenergic receptors are that they are located only in the sympathetic system, as is the case with all adrenergic receptors. These are the most abundant receptors of the sympathetic nervous system. These are usually excitatory. And as we speak about excitatory, I often have students confused when we speak about excitatory versus inhibitory. So when we talk about an excitatory response that's brought about by a receptor, this is usually a response that increases function. So for instance, if we're talking about smooth muscle, this is usually going to constrict or contract that smooth muscle. When we talk about an inhibitory response, that is going to be relaxation of that smooth muscle. So make sure you're keeping that straight or keeping that distinct as we talk about the excitatory response on the tissue. So alpha adrenergic receptors usually bring about an excitatory response, meaning that they usually cause smooth muscle constriction or increase glandular secretion, so just increase in the function of that affected tissue. Alpha receptors typically have a greater affinity for norepinephrine than epinephrine. So when we speak about affinity here, we're talking about what does this receptor prefer? Now, alpha adrenergic receptors typically prefer norepinephrine, right? Norepinephrine is going to be the more abundant neurotransmitter that's released, or a hormone, right? Remember, we're talking about either a hormone or a neurotransmitter, and both can bind to these adrenergic receptors. But the norepinephrine is what our adrenergic receptors prefer, specifically our alpha adrenergic receptors. Okay, let's move on to speak about beta adrenergic receptors. So beta adrenergic receptors, remember we've got beta 1, beta 2, beta 3. These behave very differently. So unlike the way that we classed our alpha receptors, both alpha 1 and 2 together, we're really going to look at the unique behaviors of beta 1, beta 2, and beta 3 because they behave very differently. They have different affinities. So all beta 2 receptors use cyclic AMP as their second messenger. That's kind of the first unique feature of beta receptors. And then they have differing affinities, which I'll go on to talk about, right? So the unique feature here is that they all use cyclic AMP, and I'll go on to describe what I mean by second messenger and where exactly cyclic AMP comes in. But they all use that as their second messenger. Now, beta 1 receptors have a unique location. These are found only in cardiac muscle and only in the kidneys. These are usually excitatory, so they're usually going to constrict cardiac muscle or cause contraction of cardiac muscle. And then they have equal affinity for norepinephrine and epinephrine. So we're going to contrast this to what we saw with alpha receptors. Alpha receptors prefer norepinephrine. our beta receptors have an equal affinity, meaning that they really don't have a preference. They bind norepinephrine and epinephrine with equal affinity. Beta-2 receptors found on some blood vessels and then on smooth muscle, okay? So usually the smooth muscle that lines these blood vessels in some locations, not all, is where we have beta-2 receptors specifically. We also see beta-2 receptors on the lungs. I don't know why it's missing here, but make a note of this in your own PowerPoint. Beta-2 receptors are also found on the lungs. So some blood vessels and then smooth muscles of the lungs, these are usually inhibitory in their response. So they're usually going to cause relaxation, as I alluded to earlier. So inhibitory response is going to be relaxing of that smooth muscle. Specifically in the lungs, we see relaxing of the bronchial smooth muscle, which is a part of a sympathetic response. That's going to help us to get more oxygen to come into the lungs. And then this has a greater affinity for epinephrine as opposed to norepinephrine. So again, we're seeing a difference in the way that beta-2 behaves in terms of which of these two neurotransmitters or hormones it prefers. So it prefers epinephrine in contrast to norepinephrine, okay? And then our last receptor here, beta-3 receptors. Now, beta-3 receptors are only found in fat tissue, so only in our adipose tissue. Again, usually excitatory, so usually causing contraction or constriction of these tissues. And then this also has an equal affinity for norepinephrine and for epinephrine. All right, so these are our adrenergic receptors. These are how they behave differently, and these are how they exhibit different affinities for norepinephrine and epinephrine. Now, what's really nice as a study tool, and I think you guys should do this as a homework assignment, is to create a study table where you list the receptor, so whether it's beta-3, beta-1, alpha-1, alpha-2, list its unique location, so where exactly it's found, list whether it's excitatory or inhibitory at the tissue level, and then list what its affinity is for norepinephrine and epinephrine, whether it has equal affinity, whether it prefers norepinephrine, or whether it prefers epinephrine, right? That's gonna be a really nice study tool to kind of set these receptors apart. All right, how are we doing with questions? Any questions here yet? Okay. All right, looks like everyone is doing good. So those are our receptors. And I want to take a break here to kind of really tie this into what we spoke about with regard to the general function of these two divisions of the nervous system. So when we speak about the sympathetic division of the nervous system, we speak about that fight or flight response. It's crucial to have these types of slow metabotropic receptors. That is going to help bring about a diffuse response as well. So we've got this norepinephrine flooding the system, and because of the unique locations of these adrenergic receptors, we are going to bring about different responses, which is what we want in a fight or flight response. Remember, we don't want to have all of our tissues necessarily bringing about the same effect. And if we think about that on a bigger level, think about you running from a threat, right? Running from a bear or a lion or in a fight or some type of threat. You want your digestive system to slow down because that's not the effect that you want on those tissues, but you want things like your skeletal muscle to speed up their action, to increase their metabolic activity. You want your lungs to increase the diameter of the bronchioles so that you can bring in more oxygen. You want your heart, the cardiac muscle to speed up its rate of contraction. So you want different responses on different tissues, and that's why you've got these metabotropic receptors involved in the sympathetic response, so that you can tone some functions down, but then you can tone some functions up. You can shunt blood, so we'll see that you can shunt blood towards your skeletal muscle, and away from your glands and your GI system, because that's not the tissues that you want to be vascularizing at that time. You really want blood to be shunted towards your skeletal muscle to help you escape that threat, or to fight or flight or freeze or whatever, okay? So some of our vessels will constrict their smooth muscle, and some of our vessels will relax their smooth muscle accordingly, okay? So it's a really important connection to make with these receptors and then the overall function of the sympathetic system. Okay, so I got a question here. Let's see. So do the alpha 1 and alpha 2 receptors have the same function, unlike how each beta receptor has its own unique function? That's a really good question. So the alpha 1 and alpha 2 receptors generally have the same types of function. And so that's exactly why we class them together. They're usually excitatory, usually found on the same types of tissues. The only difference is what we're going to move on to speak about now, and that is their mechanism of action. That's a type of second messenger that they use. So alpha 1 uses an entirely different second messenger action than alpha 2, whereas we saw that all the beta receptors used cyclic AMP as their second messenger. Okay, so it's a really good question. Let's see if we got any more. Okay, no. Okay, so we'll introduce this here. So this is basically the signal transduction mechanism for the sympathetic nervous system. And as I was mentioning just a minute ago, this is the unique set or the unique sequence of actions for alpha 1 receptors only. So this is what's going to happen when norepinephrine binds to an alpha 1 receptor. So you can make a note in your slides that this is only what happens. So you can make this an alpha 1 receptor. This is only what happens to an alpha 1 receptor. And so norepinephrine binds to this receptor. This is coupled to a G protein. This is something we described earlier. And that G protein has a configurational change where it changes its shape so that the alpha subunit, one of the subunits of this protein, dissociates off and goes downstream in the membrane and binds to this second protein here. Now, the second protein is then going to catalyze the reaction to split this molecule called PIP2. PIP2 is then split into IP3 and DAG, okay? So IP3 is a part of this molecule, and the enzyme that brings about this catalytic reaction is called phospholipase C, okay? So let's backtrack here a little bit. So we've got this alpha subunit dissociating off. It's going to activate this enzyme in the membrane that's called phospholipase C, and phospholipase C is going to cleave this other molecule called PIP2 into two parts. It's going to cleave it into IP3, and then the second part of this is called DAG, and I'll move on to talk about DAG in a minute. So IP3 is our, oh, here we go. So IP3 and DAG. So PIP2 is cleaved into IP3 and DAG by phospholipase C. Now, DAG is our main second messenger here. So DAG is going to enter the cell. It's going to bind to protein kinase C, which is another enzyme in our cell itself. That is going to turn on or phosphorylate some other protein in the cell, and that is how we bring about our cellular response, okay? So we describe this metabotropic type of receptor very vaguely in our previous unit, and here we're just adding on some additional detail. So this DAG is acting as our second messenger. And when we say second messenger, this is the molecule that's going to go into the cell. It's going to turn some things on. In this case, it's going to turn on protein kinase C. Protein kinase C is going to phosphorylate a protein. Anytime we see a kinase in the name, kinase means it's going to add a phosphate group, which is going to change the function of this protein by turning it on. And so we're not going to get very specific beyond this point, but this is how we then bring about a cellular response. Turning on of some protein which can be very different depending on what specific cell we're in, is what brings about the response of this receptor here. Okay. Now, the second part of this response is that, oops, we also got calcium being released from the cytoplasmic reticulum. So remember, the endoplasmic reticulum or the cytoplasmic reticulum is where we have calcium being stored. So this is an important cellular organelle that stores calcium, and then the release of that calcium is another way to bring about cellular response. So as we can see here, the binding of norepinephrine to this alpha-1 receptor brings about a series of kind of different events, right? Think about this as one messenger telling another messenger to do something else, and this chain of events that then leads to response, either via release of calcium or via the phosphorylation of some protein in the cell. Okay, and this is unique to our alpha-1 receptors.